Okay, I think that is the majority of people in the waiting room. If you know anyone that is still trying to join, we do record these and we'll go back over it at the end if anyone has any questions. But we'll go ahead and get started for now. So welcome back to those of you who've been here before. I do recognize a few of those names, and welcome to those who have never been here before. We are the Marine Environmental Education Center, located out on Hollywood Beach, Florida. And unfortunately, right now, we are still closed to the public. We are a very small facility. We're working to get open, but we do not have an opening date just yet. So please keep an eye on all of our social medias to find out more information when we do get it. Otherwise, since we are closed, our goal is to always provide some really cool educational resources to our community about environmental issues, about biology issues, environmental science, marine science. So we are lucky enough to have a wide network of very awesome scientists willing to donate their time to the MEEC and to our community to teach us a little bit about different specialties. So today we are lucky enough to be speaking to Spiro Papoulis. He is going to tell us a little bit about cyanobacteria and harmful algal blooms, which if you live in Florida, a lot of you do, you know that is a major issue we have down here. And he is going to teach us a little bit about them in general, but also his specific research into the virology. So right now, everyone's camera is off and everyone is muted. It is going to stay that way throughout the presentation, just so we can most clearly see and hear his presentation. But if you have any questions, comments, concerns, if you need elaboration on anything, or if you can't hear us or see us, something technical, please throw it into that chat, or there is a button at the bottom of your screen that has a little raise hand icon. If you raise your hand, I will make it so you can unmute yourself and explain to me if there's an issue. Otherwise, I think that's about it from me. So whenever you're ready, feel free to take it away.
Yeah, well, thank you for having me. This is very exciting. I think being invited to a webinar or something like a podcast was on my bucket list. So it's like, I check that off. So yeah, today I'm going to be talking about nutrient loading and viral methylation driving the accumulation of restriction modification system bloom forming cyanobacteria. A whole bunch of words that and a mouthful that maybe seems totally random, but I want to go through this presentation and just kind of break down a couple things about cyanobacteria, about why we should care about harmful algae blooms, and why it's important to think about them in the context of their environment and their natural predators, because it helps us understand a lot about them. So first I want to start off with the cyanobacteria. So this is one of the slides that from the lab that I originally started this research in. We like to show this slide quite a bit because it demonstrates that, right, our planet is mostly water. It's something like 70% of the Earth's surface is marine habitat, and I'm sure through this webinar many of you have come to understand that the oceans are very important for our environment, right? We fish from them a lot, we enjoy swimming in them, and a lot of Earth's Earth functioning with its biogeochemical cycles. A lot of carbon dioxide goes into our oceans and gets turned into sugar by things that go through photosynthesis. And what you're able to see here is a satellite image where we're highlighting all the places where there are chlorophyll, right? Now, chlorophyll is the same, is the component in photosynthesis that makes leaves green. It makes, I have succulents here that are green, and that's chlorophyll, right? And it's not just restricted to plants, it's also bacteria, and not just eukaryotic algae, but we're talking about actual bacteria that go through photosynthesis. So this right here is one of the most abundant cells on the planet. They live in the ocean, and they fix a lot of the carbon that's out in the environment. And so just to kind of highlight this, these cyanobacteria, these photosynthetic bacteria are right here, right? What you guys are looking at right now is a phylogenetic tree of life. So this is the way to interpret this figure is in relatedness. The closer something is together, the more related they are. So, right, we're over here, mammals, and we're kind of makes sense that we're a little bit more connected to, you know, birds and fish, these larger organisms. But then if you look over here, we have plants, everything from trees to grass to, you know, pretty much everything. And then way over here, right, very, very different from the things that you know typically do photosynthesis are the cyanobacteria. And so a little bit of background about cyanobacteria, right? As you saw in that earlier image, they're everywhere. They're all across the planet in the oceans. And what we think, these are most likely the first organisms on the planet, in the planet's history, to actually, they kind of invented photosynthesis. They're the ones that originally evolved it. And so many years ago, we're talking about prehistoric time, what people were able to do is they were able to drill into sediment, and you can look at cores of rock, right? And what people were able to find was that there was a big change in the world. And this coincided with evidence that all of a sudden there was a lot of oxygen in the atmosphere. So what they think happened was, they call it the Great Oxygenation Event, was when these bacteria, these cyanobacteria, first evolved, and they first got their ability to take carbon from the atmosphere and turn it into sugar using sunlight and using oxygen, right? Or excuse me, not oxygen. They're taking carbon dioxide and they're making sugar and they're producing oxygen, right? So they're responsible for creating our planet, our breathable air as we know it, right? And they call this the Great Oxygenation Event. It's actually likely one of the greatest extinction events of all time. Oxygen to some organisms, to a prehistoric Earth, is actually can be toxic. So yeah, they shaped our world as we know it. And then we had the evolution of plants and all sorts of stuff, which is also a great topic. So the one that I keep showing pictures of is actually called Prochlorococcus. This is the world's smallest photosynthetic organism on the planet, and it has a global population of 10 to the 27th. So for those of you, right, this is a really difficult number to articulate how many of these cells there are. 10 to the 27 written in this notation means that there are 27 zeros, right? Like we say there's a billion has 9 zeros, right? So we're talking about a crazy number of these cells all over the planet, and they're responsible for fixing 4 gigatons of carbon each year. And roughly speaking, this organism on its own is responsible for, estimations vary, but it's not too crazy to say like 1 out of 5 breaths are solely because of the cyanobacterium that exists on the planet. So back to this picture, right? Why did I originally show this? Was that a lot of the reason why Earth is the way it is because of our oceans, right? We have the fish, we have the whales, we have everything in there that make the food web as we know it, but it's really these cyanobacteria laid down the foundation going all the way to prehistoric times that generated the oceans and the Earth as we know it. And so you can see there's a lot of research that shows that in this area, right around the Pacific, is this this Pacific gyre circulates water like this. This entire cloud is essentially only Prochlorococcus. Okay, and then up here is mostly responsible some eukaryotic algae and other cyanobacteria. So the biggest takeaway that I have for you guys with this is that these cyanobacteria, if you've never heard of them before, they're extremely abundant. They're all over the place. Anytime you see a puddle on the ground and there's a little bit of green stuff in there, could be a mixture of eukaryotic algae and cyanobacteria. So what we did in our lab is we like to go out and sample a whole bunch of different parcels of water, right? And what people found out was that if they stain for DNA, which is a really easy way to look for organisms, right? If you have a little bit of water and you add something that makes DNA glow, you're able to see them much clearer. And so I always like showing this picture because it really articulates how much life there is in a given parcel of water. And so these big glowing dots are bacterial cells, right? And these little ones right here are actually viruses that prey on these bacterial cells. So there's a lot of complicated ecology that's happening in just a small little bit of water. And this is kind of the premise of what we like to look at as microbial ecologists, is we like to look at the interactions not only from bacteria to bacteria and how they drive the ecosystem, but what we were a little bit more interested in too was, well, how do these viral predators also play a role in this larger context of these, you know, of our oceans, of our lakes, of our rivers? So I'm going to give you a little bit of background about this, right? We've already talked a little bit about the cyanobacteria that exist in the ocean, and commonly, right, you can throw this into one large category of phytoplankton. So these organisms take sunlight, and they're going to go through photosynthesis, and they're going to release a whole bunch of dissolved particulates that get recycled into bacteria, and both of these groups end up feeding zooplankton. Right, and zooplankton are kind of what we think about the basis of our food net or our food web, right? That zooplankton get eaten by bigger zooplankton that eventually get eaten by krill or shrimp, or little krill and shrimp get eaten by bigger shrimp, to fish, to whales, right? All the way up to eventually, you know, we also are part of that food chain. And in the ocean, the way that it's working is these organisms are driving what we've called the microbial loop, where the phytoplankton will fix a bunch of sugar, essentially feeding everyone, right? The bacteria are going to eat the little bits that come from dying phytoplankton, and this entire system is heavily catalyzed by the presence of viruses. So you imagine out in the ocean, you have 10 to the 27th cells, right? An incomprehensibly big number of cells every day being preyed on by viruses. And as the viruses prey on those phytoplankton, they kill them and they release their exudate, their internal biological material back into the ocean, where bacteria, other bacteria that don't do photosynthesis can eat it. And other zooplankton can also, I mean, directly eat the phytoplankton, but also the particulate matter. And what we call this is the viral shunt. It's this constant cycle of viruses that are preying on these bacteria that is driving this machine. And what we like to call this, and this is actually from a paper from one of the professors at UT, Dr. Allison Buchan, talks about this. If you ever want to read this review paper about that, this is, you can think of the ocean being a giant carbon pump, right? Phytoplankton are taking carbon dioxide, turning it into sugar. That sugar is getting recycled by getting eaten by zooplankton, for example, or the viruses are driving a lot of the microbial matter to essentially be destroyed, right? And in that sinking material, that detritus, ends up sinking down to the bottom of our ocean, and that's pumping carbon from the atmosphere to the bottom of the ocean. So it's really, really, really important for this biogeochemical cycling, right? It's part of where all the carbon in the world goes. It goes from the atmosphere, goes down into the ocean. And you can think of this as a sink, right? Like trees can also be a carbon sink, right? They're also doing photosynthesis. The oceans themselves, through this mechanism, are a giant carbon pump, taking it from the atmosphere, putting it down into the sediment. So this is a really, really good example of that. And truth be told, this is actually a eukaryotic organism. So I am cheating a little bit. This is not a cyanobacteria, but it's a really good way of demonstrating the ecology in the ocean. So this is, I believe it's off the coast of Britain, I think. I'm pretty sure. And what you're looking at is a bloom of Amelia huxleyi. This is a eukaryotic organism, a coccolithophore that exists out in the ocean. And they're interesting because they form these carbonate shells, right? So all that carbon that those organisms, the cyanobacteria and the algae, you took the energy from the sun, created sugar, created themselves, right, by just using sunlight and the organics around them. And eventually it ended up in these big coccolithophores through the food chain. And what we're looking at is actually the death of a giant bloom of this Amelia huxleyi. And this is a satellite image, okay? And so this is an example of that carbon getting shuttled down into the ocean because they're actually responsible for creating the cliffs of Dover. So this limestone is actually built off of the skeletal shell of these coccoliths, these carbonate shell carrying eukaryotic organisms. So just an example of, like, you know, these things that I'm talking about are very difficult to visualize because they're microbial, right? By definition, we can't see them without a microscope, but they're building things that we can very clearly see, right? They're building giant, giant cliffs that, we're like dwarfs compared to them, right? So this is happening every day. Our oceans are doing a miraculous amount of biology. And so this seems like a very big change, but there's a story in Alice in Wonderland that essentially exemplifies I think it articulates very clearly the struggle it means to be a microbe, eukaryotic or prokaryotic, right? So either a bacteria, a cyanobacteria, a eukaryotic algae, they all struggle with trying to occupy their niche, right? They struggle every day in survival. And in this story, right, you'll recognize this as, if any of you are familiar with Alice in Wonderland, right? It's actually originally from a story called, I believe it's Through the Looking Glass by Lewis Carroll. So there's a famous scene, right, where Alice is going through Wonderland and meets the character, the Red Queen. And the Red Queen is essentially explaining to Alice, in Wonderland, you have to run in place to stay in the same spot. And if you want to go anywhere else, you have to run twice as fast. And this is an excellent analogy for what microbes are going through, right? So I drew, my cute little bacteria, and what we have is a bacterial virus. And Lee Van Valen coined this as the Red Queen hypothesis. It's the analogy to this story. And the way that he had explained it was out in the oceans and in nature in general, right? That the competition, right? It's a zero-sum game between organisms where no species can ever win, and you adversaries replace losers in the struggle for finite resources. And avoiding extinction is a constant struggle of adaptation to acquire more resources, and innovations by competitors ultimately negatively impact all others in competition. So what does that mean, right? I just spent a bunch of time talking about cyanobacteria, that they are existing in the oceans. And what I'm trying to articulate is these organisms every day that are super important for the structure of our food web, that they're important for getting carbon dioxide out of the sky, right? And feeding ourselves. Every day they have to go through this struggle where they are trying to maintain their own niche in the ecosystem. And one of the things that we're very, very interested in are viruses and the way that they play a role in this, right? We know that these organisms are super important for us. We know that Prochlorococcus, for example, is one of the most abundant cells on the planet. We know that cyanobacteria are really important, and so are eukaryotic algae that also will get preyed on viruses. Everyone gets preyed on by viruses. And as we're well aware of, having to do this remotely, right? Like viruses their ecological interaction, right? They're preying on organisms is significant, right, to understanding biology. And so I wanted to take you through the anatomy, right? Because I keep showing, right? Everyone has seen the picture of the coronavirus right now, and it's this like spherical thing, right? Bacterial viruses, first off, cannot infect us, right? They infect bacteria all the time, but they can't infect us. They look very strange, actually. And they, some say that they were that they look like the land or space shuttle, right? And once I say that, I think you might not ever be able to unsee it. So a bacteriophage, right, just to go through the anatomy really quickly, is this weird alien looking virus that has DNA in this top capsid. So if you got rid of this bottom part, you know, it kind of looks like a virus. It's like spherical, sort of. And what typically happens is that these viruses all over the planet, all the time, are floating around and they're attaching on to bacteria. And I always like to show this picture because they just look really, really weird. What they literally do is they inject this DNA down this tube into organisms once they've attached. Okay, so I took you guys on a sampling of a bunch of different biology, right? And I want to bring it back to kind of the main thing of what we want to learn, which is a lot of cyanobacteria, a lot of activity in our oceans every day, super important to our existence as human beings on the planet. Every day they're getting preyed on by viruses. All right, this is just part of the biological pump that is of diversity of carbon transfer that are happening every day around us. So the key points, right, that I want you guys to at minimum take away with: cyanobacteria, they're bacteria that do photosynthesis, right? There's a single cyanobacteria that make up about 20% of the oxygen on the planet, and viruses prey on them and other bacteria, and unfortunately us and eukaryotic algae and all sorts of organisms every day. So now that and this actually might be a really good time if there are any questions that I think that maybe we want to address, or any type of clarifying points or interesting questions. I think that this would be a really good time to
we do have quite a few questions in the chat. If you're able to pull it up, otherwise I could read them off to you.
Okay, so we got, okay, are these algae only in the ocean or are they in freshwater as well? Are they different species, but both considered cyanobacteria? Okay, great question. So cyanobacteria are a giant group of bacteria that do photosynthesis. So they're everywhere. They're in freshwater, they're in the oceans, they're all over the place. And there's lots of different kinds of cyanobacteria. Prochlorococcus being one of them, but there's Cyanobacteria, Nosticales, Anabena, all sorts of different cyanobacteria on the planet that often their biology is very different, but ultimately they kind of take up the same niche, which is forming kind of the basis of a lot of the ecosystem interactions, because they are the ones that directly will create sugar. A question about global warming. So there's a very interesting hypothesis about, so cyanobacteria are very much important in global carbon cycling. So the implications of global warming and pumping more CO2 into the atmosphere, we're not sure what the effects are going to be, right? These cyanobacteria will always be here. They were here before we existed. They'll be here, I'm sure, after we exist, right? They're pivotal to Earth. Having more of them help? Conceivably, yes, right? If you have a lot more cyanobacteria eating up a lot more carbon dioxide, doing a lot of photosynthesis, you would imagine it would help. And I would hope so. It's kind of the same rationale why we want to preserve the rainforest, right? We want a lot of green stuff to take a lot of carbon dioxide out of the atmosphere. So yeah, I think so. Oh, the bioluminescence. Okay, so the question is, the colors in the picture, and I can try and go backwards. This is not bioluminescence in a traditional sense where they are doing bioluminescence on their own. What this is that when you want to go and explore microbes in a given environment, we have special compounds that will specifically bind to DNA and intercalate into the DNA and make that luminescence happen. Although technically you can make cyanobacteria luminesce because they have photosystems, which I won't get into, but this is enough to say that this isn't bioluminescence in the sense of like the glowing bacteria you guys may have heard of. This is the scientists took a little bit of water, added a chemical that will bind to the DNA, and then when they put them under a microscope slide, they'll shine a UV light that will make them illuminate. So it's more of a method and to be able to see all the type of different cells that might exist in the water. All right. Have they, so a question about cyanobacteria evolution. So when I say that the cyanobacteria have been around since the beginning, 100% they have evolved into an entire group of organisms, right? So hundreds of thousands of years ago, likely there was that one cell that probably figured it out and became photosynthetic, maybe. We really don't know. But whether it was one organism that figured it out or a couple different at the same time, the result ended up being the same, which was over the course of evolution, this photosynthetic, prehistoric bacterium diversified into the biological diversity that we see today. So they definitely have changed a lot. But what's interesting is that the premise of where they fit in the ecosystem kind of hasn't, right? They still lay the foundation of the food web. And cyanobacteria live outside of the water. Yes, there are terrestrial cyanobacteria that can just grow on things, right? They need moisture. A good example is lichen. Lichen, I believe, it's cyanobacteria, is a symbiotic relationship between fungi and cyanobacteria. So that's an example of one that they exist outside of the ocean. Okay, great questions, guys. Okay, all right. So I talked a lot of positive stuff about cyanobacteria. It's not all positive because there's some cyanobacteria that are a huge pain. And what you're looking at is a picture of one of those bacteria. This is Microcystis aeruginosa, if you guys have heard of this before. And unfortunately, they end up growing a whole bunch by farmland. What's happening is, right, every day, we have to feed ourselves. We know that adding fertilizer to farmland is very beneficial. And so what happens is that as we do that and it rains, all of that fertilizer gets washed away, gets mobilized, and ends up in the rivers and in the lakes and in the oceans, right? And what you guys are seeing here is an aerial shot of very, very dense cyanobacterial growth. And this is a photo of what it looks like up close. Like it's actually really disgusting. It doesn't smell nice. It's very viscous. It's really, really not good for our ecosystem because when you have that much growth, it can kill life underneath in the water column. So, the plants, all the vegetation at the bottom of this river are not going to get the sunlight it usually does. And it usually like kind of chokes sunlight out of them and they end up dying. So they're very ecologically destructive. And so here's another satellite image to show the magnitude. This is actually Michigan. And unfortunately what ends up happening is here's the Metro Detroit area. Here's, the northern part of Ohio. And as it rains, water will carry essentially all of the pollution, the fertilizer, everything into the Great Lakes, where unfortunately we see a giant, and it usually happens annually, blooms of Microcystis. Now, this also happens other places in the world too. Lake Taihu in China is another example where we have this phenomenon where unfortunately the all of the nitrogen and phosphorus coming in from the surrounding area generates this cyanobacterial bloom. So, and they're commonly referred to as harmful algal blooms. Realize that when I say algal bloom, it can mean cyanobacteria, which have also been called blue-green algae. And also there are eukaryotic organisms that also bloom. And I believe the eukaryotic ones are the ones that happen in Florida a little bit more so than Microcystis. But I do believe Microcystis happens in Florida as well. So lots of biomass. And what I mean by biomass is just there's a lot of there's so many of them that you can quite literally feel it, right? If you scoop them out with your hand, it'll feel viscous. They're characterized from eutrophic, nutrient-loaded environments. Remember, this is from farm runoff. And so for all the ecological reasons, right, blocking out sunlight, all that type of stuff, it can be damaging to fish, can be damaging to the vegetation. The worst reason that we do not, the number one issue with these organisms is that they actually produce, so Microcystis, it produces microcystin, and there are other compounds that it can produce, and other cyanobacteria can do this, but this is a hepatotoxin. This means that it's toxic to your liver. And again, lots of biomass, really bad for us. Unfortunately, the people of Toledo, Ohio, over here, discovered that Microcystis can sometimes be an existential threat to the safety of a city. So what happened one year in 2014 was the winds were just right. The winds were blowing the water intake valve to the lake, the water intake valve to the city, right, where the city gets all its water, sits on Lake Erie. And so what they do when these blooms happen is that they'll close the water intake to make sure they don't get contamination. Well, one year it was a perfect storm. The water valve was open, there was a storm that came in, and the bloom was also collapsing at that time. Released a ton of microcystin into the water supply, and the city was out water for several days. So huge issue. But this is where the key question of the research that we're interested in comes in, is we know that viruses, for example, prey on organisms like Prochlorococcus, on these open ocean cyanobacteria, and they're fine every day, right? Like it's going on, but they're very spread out, right? When you go out into the middle of the Pacific Ocean, you just see blue water, right? One would imagine and we can confirm because we've done the experiments to find them, there are viruses in this water. How is it so dense, right? You would imagine, right, if you put a bunch of people, right, in the wintertime, in the wintertime, we often get sick because we're in very close proximity to each other indoors and pathogens can spread. Very similar idea to this. How is it that these cyanobacteria are able to resist viral predation every day in these lakes, right? You would imagine that they wouldn't have a very easy time doing this. Again, we're imagining how are the viruses not able to take care of this pesky organism for us, right? There has to be some sort of rationale. So bacteria often can, and a lot of other organisms, but specifically, right, the bacteria and the cyanobacteria have really two big mechanisms to prevent viruses from attaching, right? One is by physically stopping it, and then if they do get in there and they do attach, they have other ways to prevent infection, which is destroying the DNA. And I'll elaborate a little bit more about this. So what you're looking at the right here is the cell surface of a bacteria. And here's a virus coming along that wants to infect this bacteria, reproduce itself. And there's a lot of very well-studied cell surface proteins and other compounds. And this flagellar protein is responsible for, it spins around and it's like a propeller. It can get bacteria to swim through the water. And so the bacteria, this bacteriophage can exploit these things that the bacteria uses to collect nutrients or to move around. It can exploit it by literally attaching on and injecting its DNA into the cell. And the way to think about this is like a lock and key, right? And I don't want to oversimplify this. When I say lock and key, right, remember, organisms evolve all the time. Think of it like it's a changing lock and key, right? Like from the perspective of a bacteriophage, it has to float around and find just the right entrance into the cell. So it's searching this. And this is this idea of the bacteria wants to prevent this as much as possible, right? These are drivers of evolution that keep things changing over and over again. Okay, so this is now getting a little bit more specific to the research that I had done. Lots of mechanisms that can prevent bacteriophage from getting into the cell. We're going to ignore that. Let's pretend a bacteriophage actually gets in, attaches onto the cell. Now its next job, what it's doing in this cute cartoon, is the capsid protein, the top where all the DNA is stored inside of the virus, is getting spooled in through this tail protein into the bacterial cell. Remember, it's taking advantage of something on the cell surface that allows it access, right? Again, this lock and key concept. The bacteria, though, and we don't often think of them this way, but they do have immune systems, right? They're just a little bit different than our immune systems. Our immune systems, we have things like white blood cells that will go engulf bacteria, right? In this case, for bacteria, there are proteins that float around inside of the bacteria that will recognize the DNA of the bacteriophage and chop it up. So in this picture, this restriction enzyme, this Pac-Man with teeth, recognizes a specific element in the bacteriophage, and it will cut up the DNA. If you cut up the DNA of the virus, you stop infection. So this is actually I always like showing these pictures because, again, we're talking about something that is smaller than a bacteria, a protein. What we end up having to do is reconstruct what these proteins look like with computers by understanding how the protein folds together and actually does the mechanism, right? And so this bacterial immune system, these restriction enzymes or restriction endonucleases, I'll say them interchangeably, you can really think of them like DNA scissors, okay? And as a fun fact, these proteins ended up laying the foundation for the ability for us in the lab to manipulate nucleic acid. So our ability, all of these experiments where we're able to chop up DNA and analyze DNA, all of it comes from these proteins, because we would have never really been able to use DNA in the lab very easily if we couldn't manipulate them. And so these DNA scissors, these restriction enzymes, were were pivotal findings, right, in generating the technologies, the DNA sequencing technologies that we see today. So if you ever have a question about why is basic research really important, well, this is an exact example of this, where we discovered these just in bacteria, and now we use them all the time in our laboratories to run our experiments for molecular biology. So just to really concretely visualize what's happening, this protein will recognize this stretch of DNA. It's called Echo R1, and it'll recognize this sequence and cleave this DNA. And so if this is viral DNA, you know, that's it. Infection has stopped, DNA has been degraded, the bacteria has been saved. There's a second element to this system, right? These recognition sequences by random chance will exist on the bacterial genome. And when I say genome, what I mean is all of the genes, all of the DNA that make the bacteria what it is. So the genetic code, right? So if these recognition sequences are available on the DNA of the virus, they're also available in the bacterial DNA. In order to prevent accidental digestion, accidental cutting of the host, there's a second protein called a methyltransferase. And what it does is it'll add these methyl groups, the CH3, onto these recognition sequences. So a little bit more detail, what does that look like? This is originally what we saw. Echo R1 is cutting this recognition sequence. The methyltransferase comes by and throws on this methyl group. This you can think of it acting as like a block. Okay, it's like it literally, when the protein is coming on and it wants to attach onto the DNA and cut this part, right? This protein, this methyl group acts as a blocker. It'll try and come in and cut it, and it just won't happen. It's like putting it's like literally jamming scissors with like a wooden stick or something. Very, very conceptually similar. So, as a quick review, right? We started off talking about cyanobacteria, and we continued on to viruses, and now we're talking about the proteins that are responsible for protecting the bacteria from getting infected. One thing to know, and what we do know about these, is bacteria can hold on to a lot of different kinds of restriction enzymes. Okay, so it's not just one restriction enzyme, but a bunch of restriction enzymes that recognize a lot of different DNA. And the rule of thumb is the more restriction enzymes that a bacteria has inside of it, the better it is at defending against the viruses when the DNA is getting pulled into the cell. So, going all the way back to Microcystis, remember, Microcystis is blooming inside of these lakes. We're curious why it's so good at it. And specifically, we're curious on how it's able to resist the viruses that are naturally in the lakes. Like, we would expect them to never get to such dense biomass. So does Microcystis aeruginosa have, is it unique in its ability to resist viruses? So I took a lot of the way that we did this, and I would love to discuss this more if anyone has time at the end of the presentation. But effectively, what I did was we have all the microbial DNA sequence. We have a lot of it right now available. And so what I asked the question was, well, I can just analyze DNA and look for these proteins, the sequences of these proteins, and I can just count them. And so I take a bunch of DNA, a bunch of sequences that exist as data, right? And I just chug through it all in the computer. So I'm counting these bacterial defense systems in the DNA. I'm looking for these specifically to ask the question, does Microcystis have more of these? Okay, so now we're at some data. In this figure, on the x-axis, and this actually demonstrates the diversity in cyanobacteria, is our major groups of cyanobacteria, okay? On the on the x-axis. On the y-axis, we have RMPG. It's abbreviated for restriction modification per genome. Essentially, the higher this value is, the more of these DNA cutting enzymes you have inside of the cell. Now, what I did was I grouped them based on the organism, okay? And right here in the literature, they were saying that maybe organisms might have more restriction enzymes if they just have bigger genomes, right? And what I was able to show that was not the case, right? It's kind of random, right? But what we found is that, okay, well, let's look at the extremes of this. At the extremes, we have Microcystis in the yellow here. And these other yellow highlighted cyanobacterial groups, these bacterial genera is the level of classification. They're all bloom-forming cyanobacteria. So they have a very similar ecology. On the other side of this figure is Prochlorococcus and Synechococcus. Now, we mentioned a little bit more about Prochlorococcus earlier. This is the guy that's out in the oceans fixing carbon every day that exists, in a totally different environment. So I'm going to get rid of all the guys in the middle right now because it's a lot of ecology to go through. And so we're going to focus on these open ocean cyanobacteria and these bloom-forming cyanobacteria. So the ones in the green are from an oligotrophic environment. And what an oligotrophic means is just very low nutrient availability. like the open ocean. Again, we saw other pictures of these eutrophic organisms. These organisms that are coming from very, very enriched environments. So high nutrient availability from farm runoff. And this is what's generating these harmful algal blooms. And again, to reinforce this idea about what these environments look like, my PhD advisor actually, this was a picture that he took off the coast of Hawaii before a research cruise, right? And you're looking at a population of Prochlorococcus and Cyanobacteria, right? So very clearly we can see that they're very, very low abundance, right? They're there in the ocean, they're just not as dense. On the contrary, this is a picture of Microcystis taken by another professor, I believe Steve Wilhelm at the University of Tennessee, right? I think that this is off the coast. This is, he took this picture while at Lake Erie or Lake Taihu, I don't remember which one, but it doesn't matter because they both end up looking like this anyway. Very dense biomass, very, very polluted water. Again, this is the aerial shot of that, right? I mean, that's a lot of cells existing in such a small little bit of water, right? We're talking about Prochlorococcus occupies the ocean. Now, these organisms just grow up to really high biomass in just these smaller bodies of water. So this was a very compelling, a very compelling observation, right? That we have these proteins that seem to be, right, Prochlorococcus and Cyanobacteria still have these immune systems, right? But not to the extent of these other organisms. So what we hypothesized was that the environmental nutrient levels are controlling the selective pressures for restriction modification. In other words, a lot of resources are pushing these organisms to hold on to a lot more of these immune systems, a lot more of these endonucleases. And so the way that we tried to investigate this hypothesis further, right? Because we're scientists, right? We want to make sure that we don't just like, we notice this and this is what we think it is. We wanted to try and investigate this a little bit more closely, right? And the route that we took to do this was through mathematical modeling. So the idea is that if you make a mathematical model that are emulating the really important characteristics of this hypothesis, that nutrients are coming in, that they're causing the bacteria to grow up, that they are then getting preyed on viruses, you can start to squeeze a little bit more of a fine-tuned hypothesis out of what you're looking at. And I'll try and demonstrate this through a pictorial representation of what this mathematical model would look like. So we have some sort of supply rate of nutrients coming into our hypothetical environment. It's consumed by two members. What I'm going to call this competition specialist, this organism that's really good at eating the nutrient, and another organism, what I'm going to call a defense specialist, that has twice as many restriction modification systems than this competition specialist. However, and it's better at defending against viruses. However, it comes at a cost, right? If it's producing these extra proteins, it will not be as good at eating the nutrients. It won't be as good as assimilating it into biomass. And of course, we have a virus, okay? And so what we did was we used this mathematical model to try and understand at an ecosystem level what's happening as we pump more nutrients into the environment. So we have some sort of nutrient supply rate. So the amount of nutrients that are getting pumped into the system. And on the y-axis, what we have is the abundance of cells, the abundance of the green guy, the abundance of the yellow, the abundance of the virus in the dashed black line. And so what our model is suggesting to us is that as we're putting way more nutrients into the environment, the population will shift. It'll shift, this guy will grow more and more and more dense up until a certain point where the viruses come into this system, right? And the interesting part is that the viruses are keeping this competition specialist in check, okay? However, this defense specialist that's not as good at getting resources and making biomass it's only viable much later. It needs a lot more nutrients to even exist in our system. But as we pump more and more resources in there, it grows, it overcomes this competition specialist, and at a certain point, this guy goes extinct, right? It's completely eliminated from the system. There's too much virus there, and the defense specialist occupies as the primary organism. I'm running low on time, so I'm not going to go too much into this, but just to say very quickly, what we ended up finding out was by implementing the biology a little bit more in description into the mathematics, what we were able to show and suggest was that the reason why Microcystis has, almost 4 times, we're talking about having 1 or 2 restriction modification systems in these guys to having almost 15, right? A ton more of these proteins. What we were able to find out within the mathematics through these models was that there's this kind of memory mechanism happening where when a virus goes and infects a cell, it can adopt the methylation pattern and protect its own DNA. So what ends up happening, I guess I will try and go into this really quickly now, because this was probably my favorite part of this research was that these viruses, because they adopt the methylation pattern, they quote unquote remember the organism that they last infected. So with this guy, he's going to be really, really good. The one with the methylation is going to, shown with these solid lines, are going to be really good at infecting organisms that don't have the endonuclease and organisms that do, while those that aren't methylated, it's going to struggle infecting these ones with the restriction enzyme. But whenever they infect one that doesn't have it, they forget, they lose their methylation pattern. So the quick takeaway from this, and I apologize because I was running out of time, virions, the viruses, through the restriction modification systems, remember the last organism they infected, and this methylation resets every time they infect an organism. What this means on a higher level is that the diversity of these immune systems ultimately will foster coexistence between a lot of different organisms. So Microcystis is incentivized in a way by the viruses to collect as many different restriction modification systems as possible, as many immune proteins as it can in order to survive, right? And so when we go and we look out into these ocean or into the lakes that have these harmful algal blooms, right? What we're starting to put together is that the reason why these guys are actually able to grow up to such a large biomass is because they've heavily diversified the number of immune systems that they have within their cells. So when a virus infects one cell and breaks apart and it's going through, it's not going to be able necessarily to infect the rest of the population as easily. We're in a very low diversity population. That's not going to be the case. If you had just one immune system shared among all the bacteria, well, the only thing that's going to make one individual organism better is going to be having more of the restriction modification systems. But by diversifying, we end up finding that a lot of these organisms can exist together and take advantage of higher resources. And so we think that this is what's driving the harmful algal blooms. Really apologize, I was running low on time, and we can talk about this more. But I do want to really quickly acknowledge all of this work was done at the University of Tennessee. So what we do a lot of times is we actually go out to the ocean, we take our samples, or we go out to Lake Erie, and we collect these samples and we bring them back to the lab. So even in a landlocked state, we're still able to really do a bunch of really cool research. So the microbiology department, if anyone is ever interested in microbial ecology, I highly recommend checking this department out. They do a lot of really cool research. And these are going to be the committee members that were a part of this research. So Eric Zinzer was my advisor, studies Prochlorococcus. Stephen Wilhelm is the guy that primarily studies harmful algal blooms. David Talmy is is the mathematical modeler. Scott is, he helped out a lot with the computer science, and Jeff Morris is another member from the University of Alabama that studies evolution. So a lot of work went into this, and I'll try and tackle some questions right now. And of course, this was funded by the National Science Foundation. So thank you everyone for, paying taxes and having that go to the National Science Foundation. Really, really good. All right. Okay. I'm going to try and burn through a couple of these questions. So how did you decide to study this? The reason why we decided to study this was, if we're going to really try and understand these harmful algal blooms and what makes them successful, the viruses to us seem like a very good way of trying to understand their ecology. By understanding their ecology and the way that they're being so successful, hopefully we can come up with better solutions for mitigating harmful algal blooms. There's also a little bit of an insider story on this, which is that we wanted to genetically manipulate Microcystis so we could run really good experiments trying to understand the genetics of it. It turns out, when you're trying to do molecular biology on an organism that has a bunch of bacterial immune systems, it's really, really hard to do because you can't get it to take DNA up, right? So if you're ever, a common thing in microbiology is knocking out genes and testing, well, if I knock out this gene, what does it do, right? What does it have the effect on the organism? We can't do that with Microcystis because of these proteins. And that's kind of like the rabbit hole, how it ended up happening. Okay. Do you have any advice for those who are interested in this sort of microbiology field? Should they be building up any particular skill set to prepare? Okay. I will say 100%, the number one thing that you need is curiosity. Okay. So definitely there's, what I would say is continue just studying science, right? Studying, going to your biology classes, learning about microbiology. All of the biology helps understanding problems like this, right? We went from talking about, the global carbon cycling of Earth that relies on this, a couple organisms or majority, some cyanobacteria responsible for it. And we use computers, we use mathematics, we used a whole bunch of stuff. Microbiology is a wonderful science because it really pulls in ecology, evolution, biochemistry. As long as you're interested in something, as long as you find some part of science interesting, there's a place for it in microbiology. And I guarantee that it will be useful in some sort of research question, trying to learn about some sort of microbial ecology. So if you have the curiosity, that's like the most important part. Oh, okay, I'm gonna try, hang on, there are some.
It's totally fine if we have quite a few questions in the chat. Take your time answering them. We are recording this session, so if people have to start jumping off, go check out our YouTube page after it's over.
So you have a lot. Okay. So would identifying and propagating viruses that are effective against cyanobacteria help procure HABs? Very interesting. People had this idea that they thought, what I recommend is looking up this thing called phage therapy. People thought that they could use viruses actually to stop bacteria from infecting us. So far, it hasn't proved to pan out successfully. They tried doing it a lot because the idea is that instead of antimicrobials, right, instead of antibiotics, you could just give people bacteriophage that won't hurt them and it'll kill off the infection. That hasn't really worked. And I would say from what's really tough is that because there's so much biodiversity out in the ocean and in these harmful algal blooms, right? They're always going to be better at if we go in the lab and we design the perfect virus, maybe it'll work for like a little bit, but there's always going to be another subpopulation that comes up being resistant. And that's that idea of the running in place, this Red Queen hypothesis, where organisms are, it's an engine of diversity that keeps pumping out a lot more different variants. And maybe one day someone might be able to figure it out, but it's a tough one. Okay, so the different cyanobacteria. Okay, so there's a question out there about, okay, so a virus is learning multiple systems, right? If you have, and I did gloss over this very quickly, so I apologize. In this picture, right, this bacteriophage, this virus remembers this guy, okay? If this guy had a bunch of other restriction modification systems, it would be different ones that target different DNA. Actually, the best way to show this might be, good question, good catch, whoever that was. So this is an example of one cut site, okay? And I think there are a couple other questions about this. So one cut site in DNA. These restriction enzymes, EcoR1 is not the only one. There's hundreds of them and probably thousands out in nature that all recognize different DNA. The memory that I'm talking about is the memory of the virus to defend a particular site when it gets methylated. So while, let's say, a virus got propagated through a cell, infected a cell that only had EcoR1, now the virus can protect against EcoR1. But if there's another restriction enzyme that it that is trying to defend against, it won't work. Now, the idea of having a lot of different systems between a bunch of different organisms is that it's harder for the phage, the bacteriophage, to remember all of the restriction enzymes if everyone has different ones. So it's always faced with a new challenge. And that's what I was trying to get about the diversity question. Because there are so many different immune systems out there in these bacteria, it's very, very difficult for the viruses to be a catch-all and be able to infect all of them at once. All right. So there's also a question of if we know that the major cause of HABs are fertilizers in agriculture, are there safe options farmers can use or policies in place to prevent certain chemicals from being used in these products? Very good question. So it's a struggle, right? We need to make crops and we want them to produce a lot of, we want to produce a lot of corn, or we want to grow things to make sure we can all feed ourselves. So what has worked in mitigating HABs is by limiting the amount of fertilizer that we're putting on the farmlands, which has definitely helped out a lot. Now, I can't really speak necessarily because I don't really know the policy stuff with the fertilizers that are allowed, but generally speaking, some fertilizers that are a little more recalcitrant, so they're not as soluble by water, are always going to be a little bit better because you want to minimize the amount that are getting run off. So one example is like plants need phosphorus, right? And they can get it from certain minerals that as the plant roots are growing, they end up solubilizing a little bit more of these minerals that they need. So I mean, I don't know. I'm personally, I'm not actually an expert on fertilizers themselves, but definitely we know the link with fertilizers, and there have been efforts to try and minimize the amount, not putting it on an excessive amount, because it does have very negative consequences. And we have lots. So you said we have lots of DNA mapped. What percentage has been completing? I'm going to interpret this as how many microbes we've sequenced. So we've sequenced. So in order to analyze bacterial DNA, we need to have microbes themselves sequenced. And as of Oh, I think as of last year in our public repositories, right? So the federal government will give money to the NIH, the National Institute of Health, and they also, right, in the NSF to fund scientists' research. What they also give to, they have a specific program called NCBI, it's National Consortium of Biotechnology. It's essentially a big thing that the government does that stores all these sequences. So if you go to NCBI, and this actually might be fun just to do real quick. So if you Google NCBI, the National Center for Biotechnology Information, that's what I was looking for. Every single scientist, every biologist that does anything with DNA, this could include ones that work at companies or ones that work within their universities, all use NCBI as a resource to look at DNA. So the human genome is stored here. 180, I think it's 180,000 as of, I think a year ago, 180,000 microbial genomes are stored here. This is terabytes and terabytes of data that is freely available for anyone to go look up, right? And so you can look up articles, all sorts of stuff, but you can also look up genes and what they do. You can look up protein sequences. It's a fantastic resource. And as we collect more of this information, there'll be a lot more, hopefully there'll be a lot more research into trends within this data, right? Because to be clear, I did not sequence any of the genomes that I analyzed. Everything that I analyzed was from NCBI. So this is an exceptionally powerful public good for the research community. And I definitely I think this is a really good example of where the funding for research really helps it really is a public resource for the entire research community to be able and look up these genomes. So hopefully that maybe I hope I answered that question right. All right, I'll stop my share.
I think that you hit most of the questions. If we missed anyone's question, or if you think of one later on, feel free to email us or direct message us on any of the social medias for the MEEC. Otherwise, our email is meec@nova.edu. I definitely won't be able to answer those questions because it is above my head, so I will send them right on to Spiro and we will get back to you. But thank you so much. That was an awesome presentation. I learned very, very good.
Yeah, any questions, guys, feel free to ask. I mean, I definitely, especially if people are interested in microbial ecology, there's a ton of research out there, not just about harmful algal blooms, studying the human microbiome, studying organisms microbes that live on plants, all sorts of stuff. I mean, it's astrobiology, people are interested in microbes in space. There's a lot out there. So if you have a curiosity, chase it.
Brilliant. So thank you all for coming today. I hope you enjoyed the presentation. And if you missed any of it, we do record it. It does have a bit of a slow turnover rate to get onto our YouTube channel. Sorry about that, but I will get it up there as soon as possible. So make sure you look us up, Seek the MEEC on all the socials and YouTube. And we are continuing this webinar series. So next Saturday at 1 p.m., I believe that's the 15th, we are having Joanna Figueroa coming in and talking to us about coral spawning and recovery. So she is going to get a little more technical. I like this month, we have a lot of our heavy hitters coming in, teaching us the technical aspects of science. So she is telling us a little bit about coral spawning specifically and what they're doing in the lab to sort of help that along and help with recovery efforts. So it is the same link. If you are free next Saturday and want to check that out, feel free to come join us then. Otherwise, thank you all for coming today, and thank you very, very much for presenting today. And everyone stay safe out there.